All right, let's dive right in. We're going to tackle one of the most fundamental questions in all of biology. How does the genetic blueprint, our DNA, actually get up and build something? How does it create the unbelievably complex machinery of a living cell? Well, the answer lies with a truly remarkable molecule that's at the very heart of it all.
I mean, think about it for a second. A blueprint for a house? It's just paper. It can't pour the concrete, it can't raise the walls by itself, it needs a construction crew, it needs working copies, it needs tools to make any of that a reality. And in the tiny world of our cells, DNA is that master blueprint, and it's locked away in the nucleus for safekeeping. That construction crew we're talking about, the star of our show today, that's RNA.
So, in this explainer, we're going to take a 5-step journey together. First up, we'll meet RNA itself, this dynamic multi-tool of the cell. Then we'll see how it actually copies the genetic code. After that, we'll uncover this really surprising editing process it goes through. Then we'll follow it out to the factory floor where it builds a protein. And finally, we'll explore how understanding RNA is literally changing our world.
Okay, so let's start with our main character. You see, unlike DNA, which is built for super stable, long-term storage, RNA is built for action. It really is the cell's indispensable utility player, capable of wearing a ton of different hats.
A few seemingly small chemical tweaks make a world of difference here. DNA, as you know, is that stable double helix. It's perfect for being an archive. But RNA is single-stranded, and that lets it fold up into all these complex 3D shapes. It also swaps out one of the bases, thymine, for something called uracil. These changes make it the perfect working copy. Yeah, it's less stable than DNA, but it's way more versatile and totally ready for immediate use.
Now, to tell this story right, we need to meet the main cast. First, you've got messenger RNA, or mRNA. This is the molecule that actually carries the gene's instructions. Then there's transfer RNA, or tRNA. Its cool cloverleaf shape makes it the perfect little delivery truck for bringing in the right building materials. And finally, there's ribosomal RNA, or rRNA, which actually folds up to become the core of the factory itself, the ribosome.
So how does this whole process even kick off? Well, the very first step is getting those instructions out of the secure main office, the cell's nucleus, where the DNA is kept safe. And this process has a name: transcription. This whole thing follows a rule that's so fundamental to life, it's called the central dogma of biology. Basically, information flows in one direction, from the permanent storage of DNA to the temporary message of RNA, and finally to the functional end product, a protein. This is the core logic we're going to be following.
The engine driving this process is an enzyme called RNA polymerase. Just think of it like a master scribe. It finds the right spot on the DNA, unwinds a small section, and then meticulously creates a temporary RNA copy of a single gene. This is our first draft of the message. But here's the thing. That first copy isn't ready for prime time, not even close. Just like a writer's first draft, it has to be edited and polished up before it can be sent out. And this post-production phase is absolutely crucial.
You see, this initial copy, which we call pre-mRNA, is actually a mix of useful instructions, called exons, and these long, non-coding stretches called introns. So to create a clean, coherent message, all of those introns have to be precisely cut out. This whole editing process involves 3 key steps. First, a protective cap, kind of like a little helmet, is added to the front of the message. Then comes the most important step. The introns are spliced out. Finally, a long tail, called a poly A tail, is added to the end, which acts as a stabilizer and also helps it get exported out of the nucleus.
So, what kind of machine performs this incredibly delicate surgery? It's called the spliceosome. And here is the really amazing part. The spliceosome itself is built from a special class of RNA molecules called SNP-RNAs. That's right, it is RNA editing RNA. It's just a beautiful demonstration of how incredibly multifaceted this molecule really is.
And the story gets even more complex. There's a process called alternative splicing that can mix and match the exons in different ways, and a staggering 95% of our genes use this trick. This means one single gene can be spliced in multiple ways to produce a whole family of different proteins. It's a huge source of our biological complexity.
So, our mRNA message is now edited, it's capped, it's got its tail, and it's finally ready to leave the nucleus and head out to the factory floor, the cytoplasm. This is where the instructions are finally translated into an actual physical product, a protein. To do this, the cell uses a universal language,
the genetic code. The ribosome reads the bases on the mRNA in groups of 3, and these are called codons. Each codon specifies one of the 20 amino acids, which are the building blocks of proteins. For example, the codon AUG always says, "Start here," and add the amino acid methionine.
Now the ribosome, which remember is made of our friend rRNA, is the factory. It has 3 key workstations. You have the A site for arrival, which is where a new tRNA delivery truck docks with its amino acid cargo. Then there's the P site, the workshop where that amino acid gets added to the growing protein chain. And finally, the E site for exit, where the now empty tRNA truck leaves.
And this all happens in this beautiful, repeating, 3-step cycle. A tRNA arrives and decodes the mRNA codon. The ribosome then creates what's called a peptide bond, adding the new amino acid to the chain. And then the whole ribosome shifts or translocates one codon down the line, ready for the next tRNA. It just does this over and over, building the protein one amino acid at a time.
So we've just seen the entire journey from a DNA gene to a functional protein, all made possible by RNA. But the story doesn't end there. Not anymore. By understanding these fundamental processes, we've actually learned to speak RNA's language, turning it from just a cellular messenger into a revolutionary tool for medicine.
And the impact is, well, it's on a global scale. We're talking about billions of mRNA vaccine doses that have been administered worldwide, a multi-billion-dollar market for RNA-based therapies, and thousands of clinical trials that are underway for technologies like CRISPR, which itself relies on a guide RNA to work.
Each of these incredible technologies cleverly hijacks a natural RNA process. mRNA vaccines, they just give our cells a temporary mRNA message to build a viral protein, which then trains our immune system. RNA interference therapies use tiny RNAs to find and silence the genes that cause disease. And CRISPR uses a guide RNA to direct an enzyme to a super precise spot in our DNA for editing. We are literally reprogramming biology.
So you see, RNA is so much more than just a messenger. It's a scribe, it's an editor, it's a factory worker, and now, in our hands, it's one of the most powerful tools we have to engineer biology and to fight disease. It really leaves you wondering, what problems will we solve, and what new things will we build by harnessing the power of this incredible molecule?